Hey guys, welcome to another episode of the Human AI podcast. Today we're going to talk about celiac disease and inflammatory bowel disease. If you're unfamiliar with how the structure of the podcast goes, I'm just the host that talks about these topics, but I don't know them, so I have AI with me, which is our guest. In this situation, Grok is going to be explaining to us these topics today. And we will take everything Grok says with a pinch of salt, as it might be inaccurate at times. So, cross reference everything you hear to proper science, and without further ado, let's get into today's topic. Grok, let's start with celiac disease. What is celiac disease?
Celiac disease is an autoimmune disorder where eating gluten, a protein in wheat, barley, and rye, triggers an immune response that damages the small intestine's lining. This leads to nutrient absorption problems, causing symptoms like diarrhea, bloating, fatigue, and weight loss.
Interesting. So it's an autoimmune disease. It's gluten sensitive. It's got to do with the intestines, with the GI. Gluten is a protein. Okay. So what are the genetic abnormalities of celiac disease?
Celiac disease is strongly linked to specific genetic markers, primarily the HLA-DQ2 and HLA-DQ8 genes, which are present in about 95% of patients. These genes influence immune response, making the body react abnormally to gluten, though having them doesn't guarantee the disease, as environmental factors also play a role.
So what is chromosome 6p21 and how this is linked?
Chromosome 6p21 is the region where the HLA genes, specifically HLA-DQ2 and HLA-DQ8, are located, which are critical in celiac disease. These genes code for proteins that present gluten fragments to immune cells, triggering an autoimmune attack on the small intestine in genetically susceptible individuals.
And what about people with Down syndrome, type 1 diabetes, and autoimmune hepatitis? Are these people more susceptible?
People with Down syndrome, type 1 diabetes, and autoimmune hepatitis are more susceptible to celiac disease due to shared genetic and autoimmune predispositions, particularly involving HLA genes. Down syndrome increases the risk about 5-10 times, while type 1 diabetes and autoimmune hepatitis patients have a 5-7% and up to 20% chance, respectively, of developing celiac disease.
Okay. And how does gluten cause celiac disease? What does gluten do with the small bowel mucosa? Talk to me about transglutaminase, diamidates, glutamine and gliadin, interleukin 15. What's going on?
Gluten, specifically its gliadin component, triggers celiac disease in genetically susceptible people by interacting with the small bowel mucosa. When gluten is ingested, tissue transglutaminase, an enzyme, not glutaminase, deamidates gliadin, converting glutamine residues into negatively charged residues that bind strongly to HLA-DQ2 or HLA-DQ8 on immune cells. This sparks an immune response where interleukin 15, produced by intestinal cells, amplifies inflammation, activating T cells that attack the mucosa, damaging the villi, finger-like projections that absorb nutrients. Over time, this flattens the villi, reducing surface area and causing malabsorption, leading to symptoms like diarrhea and malnutrition.
So what is tissue transglutaminase and what are diamidates? What they have to do? And what is this negatively charged protein have to do? Interleukin 15 specifically? What do natural killer cells have to do? Intraepithelial T lymphocytes? And how does eventually tissue destruction occur?
Tissue transglutaminase is an enzyme in the small intestine that modifies gliadin by deamidation, turning its glutamine residues into glutamic acid, creating negatively charged proteins that bind tightly to HLA, DQ2, or DQ8, triggering an immune response. This process, not involving diamanides, likely a misinterpretation of deamidation, activates interleukin 15, which rallies natural killer cells and intraepithelial lymphocytes, immune cells in the gut lining, to attack the mucosa, causing inflammation and villus atrophy, where the intestinal villi are destroyed, leading to tissue damage and malabsorption.
Got it. Wow, that's such a big process. And the normal small bowel lining, they are like finger-like. Well, what about the abnormal?
The normal small bowel lining has finger-like villi that maximize nutrient absorption, but in celiac disease, the abnormal lining becomes inflamed and damaged due to the immune response to gluten. The villi flatten or disappear, a process called villus atrophy, leaving a smooth, scarred surface that severely impairs nutrient uptake, causing symptoms like diarrhea and malnutrition.
Okay, so what are the symptoms of celiac disease? Talk to me about how flat mucosa might not be able to absorb nutrients, and how we detect celiac disease too. Tell me the difference between classical and atypical celiac disease.
Celiac disease symptoms vary widely. Classical celiac disease presents with gastrointestinal issues like diarrhea, bloating, abdominal pain, and weight loss, caused by flat mucosa villus atrophy, which reduces the small intestine's surface area, impairing nutrient absorption and leading to malnutrition, fatigue, and deficiencies like anemia or osteoporosis. Atypical celiac disease may show suppler or non-GI symptoms, such as joint pain, dermatitis herpetiformis, an itchy skin rash, infertility, or neurological issues like ataxia, often with less obvious nutrient malabsorption. Detection typically involves blood tests for antibodies, like anti-tissue transglutaminase or anti-endomysial antibodies, followed by a small intestine biopsy to confirm villus atrophy, though genetic testing for HLA DQ2, DQ8 can support diagnosis.
I want you to quiz me on these things you just said. Just like give me 5 rapid quiz questions. I'll answer it in a couple of words, and I'll answer in a couple of words.
All right, here we go with 5 rapid-fire questions on celiac disease. 1. What protein triggers celiac disease? 2.
No,
Name
no,
one.
dude. Give them one by one. We're not in a rush.
My bad. Let's take it one by one. First question. What protein triggers celiac disease?
Gluten.
Nice, spot on. Next question. Name one genetic marker linked to celiac disease.
Those weird ones. HLA DQ2. Can you explain what they mean? Before we go to question 3, what does HLA DQ2 mean? What's this acronym?
HLA DQ2 stands for human leukocyte antigen, specifically the DQ2 subtype, which is a protein encoded by genes on chromosome 6p21 that helps the immune system recognize foreign substances. In celiac disease, HLA DQ2 binds to gluten fragments, triggering an autoimmune response that attacks the small intestine in genetically susceptible people. All right, ready for question 3. What enzyme deamidates gliadin in celiac disease?
Tissue transglutaminase.
You got it. Question 4. What's one gastrointestinal symptom of classical celiac disease?
I forgot. What, also, tell me, but why is tissue transglutaminase called tissue transglutaminase?
Tissue transglutaminase is called so because it's an enzyme found in tissues that catalyzes the cross-linking of proteins by forming bonds between glutamine and lysine residues, or in celiac disease, it deamidates gliadin, enhancing its immunogenesis. The trans refers to its ability to transfer or modify these protein groups, and glutaminase relates to its interaction with gliadin, though it's distinct from true glutaminases that break down glutamine differently. Now, back to that fourth question. What's one gastrointestinal symptom of classical celiac disease?
I'm not sure, can you tell me? Tell me, dude.
All right. One gastrointestinal symptom of classical celiac disease is diarrhea. Ready for the fifth question?
No.
Name one.
We're all good. Okay, let's talk a bit about these symptoms then. So diarrhea, smelly bulky stool, rich in fat, flatulence, borborygmus. Explain what these are. Flatulence, borborygmus.
Flatulence is the buildup and release of gas in the digestive tract, often caused by fermentation of undigested food in celiac disease, leading to bloating and frequent passing of gas. Borborygmus refers to the rumbling or gargling sounds in the intestines, caused by gas and fluid moving through the gut, which can be more noticeable due to the inflamed, malabsorptive small intestine in celiac disease.
Okay. I don't understand what borborygmus and flatulence are.
Flatulence is when gas builds up in your gut, often from undigested food fermenting, and gets released as farts, causing bloating or discomfort, common in celiac disease due to poor absorption. Borborygmus is the fancy term for those loud, gurgling noises your stomach or intestines make when gas and liquids slosh around, more noticeable in celiac because the inflamed gut moves things unevenly.
Okay. Tell me a typical celiac disease now. Tell me extra intestinal symptoms such as anemia, osteopenia, osteoporosis, itchy skin, lack of periods, pins and needles, loss of balance. Why would we have all these?
Atypical celiac disease often presents with extraintestinal symptoms rather than classic gut issues, as the autoimmune response to gluten affects various systems due to chronic inflammation and nutrient malabsorption from damaged small intestine villi. These can include anemia from iron or B12 deficiency, causing fatigue, osteopenia or osteoporosis, and confusion from calcium and vitamin D malabsorption, weakening bones, itchy skin from dermatitis herpaphoresis, a blistering rash linked to gluten, amenorrhea, lack of periods due to hormonal disruptions or malnutrition, pins and needles from peripheral neuropathy related to B vitamin deficiencies, and loss of balance from ataxia caused by gluten affecting the nervous system, particularly the cerebellum. These symptoms arise because the body struggles to absorb key nutrients like iron, calcium, and vitamins, while the immune response may directly attack tissues like skin or nerves,
That makes
even
a
without.
lot of sense. Okay, and what about emaciation? What is this? And pot belly due to gaseous distension?
Emaciation in celiac disease refers to extreme weight loss and muscle wasting due to severe nutrient malabsorption from the damaged small intestine, leaving the body starved of calories and protein, often seen in advanced or untreated cases. A pot belly from gaseous distension happens when undigested food ferments in the inflamed gut, producing excess gas that bloats the abdomen, creating a swollen appearance despite overall thinness, especially common in children with celiac disease.
I don't understand emaciation.
Emaciation is when someone becomes extremely thin, almost skeletal, due to severe weight loss and muscle wasting, often because the body can't absorb enough nutrients, like in untreated celiac disease, where the damaged small intestine prevents proper uptake of calories, proteins, and fats. It looks like a starved, gaunt appearance, with visible bones and little muscle or fat left, distinct from normal slimness.
And what's the etymology of this word?
The word emaciation comes from the Latin emaciatus, the past participle of emaciare, meaning to make lean or waste away, derived from e, out, and macies, leanness or thinness. It evolved through Old French and Middle English to describe the extreme thinness seen in conditions like celiac disease, where nutrient malabsorption causes severe weight loss.
Okay. And let's talk a bit about investigations for celiac disease. So FBC, UNEs, LFTs, what are these? Talk to me about serology, talk to me about IgA, tissue transglutaminase IgA, endomysial IgA, yeah, do biopsies.
All right, let's dive into investigations for celiac disease. First, the abbreviations. FBC is a full blood count, checking for anemia or low platelets, often seen in celiac due to nutrient deficiencies. U and E's stands for urea and electrolytes, assessing kidney function and electrolyte imbalances. LFTs are liver function tests, which can be abnormal in celiac due to inflammation or nutrient issues. Serology is key for celiac diagnosis, focusing on specific antibodies. IgA, tissue transglutaminase, TTG-IGA, is the primary test, highly sensitive and specific, detecting antibodies against tissue transglutaminase, the enzyme involved in gluten's autoimmune response. IgA endomysial antibodies, EMA-IGA, are another test, less commonly used but very specific, targeting the connective tissue around smooth muscle. Since these tests rely on IgA, total IgA levels are checked to rule out IgA deficiency, which can cause false negatives. If deficient, IgG-based tests like deaminated gliadin peptide, DGP, IgG, are used. If serology is positive or suspicion remains high, a small intestine biopsy is done via endoscopy, taking samples from the duodenum to confirm villus atrophy, crypt hyperplasia, and increased intraepithelial lymphocytes, hallmarks of celiac disease. These findings, combined with serology and clinical symptoms, seal the diagnosis, though genetic testing for HLA DQ2, DQ8 can help in unclear cases.
Okay. So tell me about the main test again.
The main tests for celiac disease start with serology, specifically the IgA tissue transglutaminase TTG-IGA test, which detects antibodies against the enzyme triggered by gluten, and checking total IgA levels to avoid false negatives due to IgA deficiency. If serology is positive or suspicion persists, a small intestine biopsy via endoscopy confirms the diagnosis by showing villus atrophy, crypt hyperplasia, and increased intraepithelial lymphocytes in the duodenum.
I don't get it. Explain it one more time very simply.
To diagnose celiac disease, doctors first do a blood test called IgA tissue transglutaminase, TTG-IGA, to check for antibodies caused by gluten, and they also test total IgA to make sure the results are reliable. If the blood test suggests celiac or symptoms are strong, they take a small piece of the small intestine during an endoscopy to look for damaged villi and inflammation, which confirms the disease.
So we do something with the IgA.
Yeah, in celiac disease testing, the IgA tissue transglutaminase, TTG-IGA blood test is key, detecting antibodies your body makes against tissue transglutaminase when gluten triggers an immune response. They also check your total IgA levels to ensure the test isn't falsely negative due to IgA deficiency, which can mess up the results.
So Just a sec, guys.
All right,
So
taking a quick...
what's going on with so IgA is IgA tissue transglutaminase?
IgA tissue transglutaminase, or TTG-IGA, is a specific antibody test for celiac disease, where IgA refers to the immunoglobulin A class of antibodies your immune system produces. It's not IgA itself, but the test that detects IgA antibodies targeting tissue transglutaminase, an enzyme that gluten interacts with, triggering the autoimmune damage in the small intestine.
Is it or is it not?
Sorry, I'm not sure what you mean by is it or is it not. Could you clarify if you're...
Transglutaminase tissue, is it the same as IgA?
No. Tissue transglutaminase, TTG, and IgA are not the same. Tissue transglutaminase is an enzyme in the body that modifies gluten, triggering an immune response in celiac disease, while IgA is a type of antibody that the immune system produces, and the TTG-IGA test detects IgA antibodies, specifically targeting tissue transglutaminase to diagnose celiac disease.
Okay.
All right, so we've been diving deep into...
Why do we call it tissue transglutaminase IgA?
We call it tissue transglutaminase IgA, or TTG-IGA, because the test detects IgA antibodies that specifically target tissue transglutaminase, the enzyme in the body's tissues that gets attacked by the immune system in celiac disease when gluten is present. The IgA part refers to the type of antibody, immunoglobulin A, being measured, and tissue transglutaminase is the antigen or target the antibodies bind to, making it a clear name for this diagnostic test.
And what are the microscopic features of celiac disease?
Under a microscope, celiac disease shows villus atrophy, where the small intestine's finger-like villi are flattened or gone, reducing nutrient absorption, along with crypt hyperplasia, where the intestinal crypts deepen due to increased cell turnover. You also see increased intraepithelial lymphocytes, immune cells infiltrating the gut lining, causing inflammation and damage.
Interesting. Villus atrophy. So explain crypt hyperplasia again and tell me about the increase in lymphocytes in the lamina propria too.
Crypt hyperplasia in celiac disease means the crypts, which are the deeper, tubular parts of the small intestine's lining, where new cells are made, get longer and more crowded as the body tries to replace damaged cells faster due to gluten-triggered inflammation. The lamina propria, the connective tissue layer beneath the intestinal epithelium, also shows an increase in lymphocytes, immune cells like T cells that infiltrate and drive inflammation, further damaging the mucosa, alongside the intraepithelial lymphocytes already attacking the surface.
Okay, so tell me a bit about complications now. Enteropathy associated with T-cell lymphoma, high risk of adenocarcinoma of small bowel, dermatitis herpetiformis.
Celiac disease can lead to serious complications if untreated. Enteropathy-associated T-cell lymphoma, a rare but aggressive cancer, arises from chronic inflammation in the small intestine, while the damaged mucosa also increases the risk of small bowel adenocarcinoma, another rare malignancy. Dermatitis herpetiformis, an intensely itchy, blistering skin rash, occurs in about 10 to 15% of celiac patients due to gluten-triggered immune deposits in the skin. These complications are linked to prolonged gluten exposure, highlighting the need for a strict gluten-free diet to reduce risks.
Tell me more about this. Can you explain it a lot very simply, please?
Okay, let's break down those complications of celiac disease super simply, like I'm explaining it to a friend. First, celiac disease, when you don't treat it by avoiding gluten, can cause big problems because the immune system keeps attacking the small intestine, making it inflamed and damaged for a long time. This ongoing damage sets the stage for some serious issues. One complication is called enteropathy-associated T-cell lymphoma. That's a mouthful, I know, but basically, it's a rare type of cancer that can grow in the small intestine. The constant inflammation from gluten makes certain immune cells, called T cells, go haywire over years, and in rare cases, they turn cancerous. It's like the immune system, trying to fight gluten, gets so overworked, it starts causing trouble itself. Another issue is a higher risk of adenocarcinoma of the small bowel. This is another type of cancer, also rare, where the cells lining the small intestine start growing out of control. The damaged, inflamed lining from celiac disease makes it easier for these cancer cells to develop, kind of like how a scratched surface is more likely to get worse if you keep irritating it. Then there's dermatitis herpetiformis, which is a super itchy, bumpy, blistery skin rash that about 1 in 10 celiac patients might get. It's not cancer, but a skin reaction to gluten. Your immune system, annoyed by gluten, creates antibodies that end up in your skin, causing these itchy patches, usually on elbows, knees, or your back. It's like your body's yelling, "Get this gluten out," through your skin. All these happen because gluten keeps triggering the immune system to attack the body intestine for cancers,
What
skin
is
for
this
the
T cell lymphoma though?
enteropathy-associated T-cell lymphoma is a rare aggressive cancer that can develop in the small intestine of people with untreated or poorly managed celiac disease. It happens when T cells, a type of immune cell, become cancerous due to long-term inflammation from gluten, damaging the intestinal lining, leading to symptoms like abdominal pain, weight loss, or obstruction.
And we are not very sure about gluten-free diet if it's actually going to reduce the risk of complications.
A strict gluten-free diet is widely accepted as the most effective way to reduce the risk of complications in celiac disease, like T-cell lymphoma, small bowel adenocarcinoma, and dermatitis herpetiformis, by stopping the autoimmune attack on the small intestine. Studies show that adhering to the diet for at least 3-5 years can significantly lower these risks, though some uncertainty remains in rare cases where damage persists or cancers develop despite compliance.
Can you reference these studies, please, and be very, very, very scientific with them?
All right, let's dive into analyzing studies on the effect of a gluten-free diet, GFD, on reducing the risk of complications like enteropathy-associated T-cell lymphoma, EFD, and small bowel adenocarcinoma and celiac disease, focusing on a rigorous scientific approach. Given the context, I'll evaluate key studies, scrutinize their methodologies, and synthesize findings, maintaining a critical eye on the data and avoiding over-reliance on any single narrative. I'll keep responses concise, as if speaking aloud, and limit to 2 sentences per message, per your preference. A pivotal study by Solano et al., 2008, followed 1,757 celiac patients over 31,801 person-years, finding that strict GFD adherence reduced the standard morbidity ratio for EFD from 6.42 to 0. That's a 95% CI of 0.02, 0.88, P plus 0.001. The significant drop suggests a protective effect, but the observational design limits causal inference, and dietary compliance was self-reported, introducing potential bias. The same study noted that only 2 of 9 EFD cases occurred in strict GFD adherence with a significant risk, X naught GWAT plus 6 naught GWAT plus P naught 1, and those with dietary gluten intake. This supports a protective role of GFD, though the small number of EFD cases reduces statistical power and calls for...
Let's jump to inflammatory bowel disease. What constitutes inflammatory bowel disease?
Inflammatory bowel disease, or IBD, including Crohn's disease and ulcerative colitis, is caused by a mix of genetic predisposition, immune system dysfunction, environmental triggers, and gut microbiome imbalances. Specific genes like NOD2 increase susceptibility, while factors like smoking, stress, or a Western diet can trigger an overactive immune response that attacks the gut lining, leading to chronic inflammation.
The main ones, it seems to me, are Crohn's disease, ulcerative disease, ulcerative colitis, and diverticular disease.
Actually, Crohn's disease and ulcerative colitis are the main types of inflammatory bowel disease, IBD, characterized by chronic immune-mediated inflammation of the gut. But diverticular disease isn't classified as IBD. It's a separate condition involving pouches, diverticula, and the colon that can become inflamed, diverticulitis. Crohn's can affect any part of the digestive tract, often with patchy, deep inflammation, while ulcerative colitis is limited to the colon, causing continuous, superficial mucosal inflammation.
Okay. Let's talk a bit about Crohn's disease. It's often complicated by fibrosis and obstructive symptoms. Tell me about it, and tell me about it epidemiology too.
Crohn's disease is a chronic inflammatory bowel disease, causing inflammation anywhere from mouth to anus, often in the ileum or colon, with patchy, deep lesions leading to complications like fibrosis, scarring, and obstructive symptoms such as abdominal pain, bloating, or bowel blockages due to narrowed gut segments. Epidemiologically, it affects about 1-2 per 1,000 people in Western countries, with higher incidence in North America and Europe, peaking in young adults, 15-35 years, slightly more common in females, and linked to genetic factors, e.g., NOD2 mutations and environmental triggers like smoking.
Okay. And what causes it then?
Crohn's disease is caused by a complex interplay of genetic predisposition, like NOD2 gene mutations, an overactive immune system attacking the gut, environmental factors such as smoking or high-fat diets, and gut microbiome dysbiosis. These trigger chronic inflammation, leading to tissue damage, fibrosis, and symptoms like diarrhea, pain, and obstructions, though the exact initiating factor remains unclear.
Interesting. Let's talk about the genetics of Crohn's disease then.
Crohn's disease has a strong genetic component, with over 200 susceptibility loci identified, the most significant being the NOD2 gene on chromosome 16, which increases risk by impairing immune responses to gut bacteria. Other key genes, like ATG16L1 and IRGM, affect autophagy, disrupting bacterial clearance and fueling chronic inflammation in the gut.
And what is NOD2 gene?
The NOD2 gene, located on chromosome 16, encodes a protein that acts as an intracellular sensor for bacterial components, helping regulate immune responses in the gut. In Crohn's disease, NOD2 mutations impair this function, leading to an overactive immune response against gut bacteria, triggering chronic inflammation.
Okay, is there a possible infectious cause of Crohn's disease?
Some evidence suggests an infectious cause for Crohn's disease, with bacteria like Mycobacterium avium subspecies paratuberculosis, MAP, and adherent invasive Escherichia coli, AIEC, implicated in triggering or exacerbating inflammation in genetically susceptible individuals. However, no single pathogen is consistently proven causative, and infections likely act as environmental triggers alongside genetic and immune factors, rather than sole causes.
Okay. And what environmental factors are implicated in Crohn's disease? Tell me about the improved hygiene hypothesis.
Environmental factors linked to Crohn's disease include smoking, which worsens disease severity, high fat or processed food diets, urban living, and stress, all of which can disrupt gut microbiome.
Keep going.
All right, so picking up on environmental factors for Crohn's disease and the improved hygiene hypothesis, reduced exposure to pathogens in early childhood due to cleaner environments, like in urban or Western settings, may prevent the immune system from developing proper regulation. This can lead to an overactive immune system later, contributing to chronic inflammation in genetically susceptible people, though the exact mechanisms are still under study.
Okay. So what exactly is it though?
The improved hygiene hypothesis for Crohn's disease suggests that growing up in overly clean environments with less exposure to germs, parasites, or bacteria stops the immune system from learning how to balance its responses properly. This can make it overreact to harmless gut bacteria later in life, triggering chronic inflammation in those with genetic risks, like NOD2 mutations, contributing to Crohn's.
And what is the evidence for this?
Evidence for the improved hygiene hypothesis in Crohn's disease comes from epidemiology studies showing higher Crohn's symptoms in developed countries with better sanitation, like North America and Western Europe, compared to developing regions. Observational data, such as a 2008 study in gastroenterology, finding lower Crohn's rates in areas with higher childhood exposure to infections, and animal studies showing altered gut immunity in sterile environments support the idea that limited microbial exposure early in life disrupts immune tolerance, though direct causal proof remains limited.
I see. Okay, now let's talk a bit about the clinical features of Crohn's disease.
Crohn's disease typically presents with abdominal pain, chronic diarrhea, often with blood, weight loss, and fatigue, due to patchy inflammation that can affect any part of the digestive tract, most commonly the ileum and colon. Complications like fistulas, abscesses, or strictures may cause additional symptoms such as fever, severe pain, or bowel obstruction, while extraintestinal issues like joint pain, skin rashes, or eye inflammation can also occur.
Okay. So, what's the distribution of Crohn's disease?
Crohn's disease can affect any part of the digestive tract, from mouth to anus, but it most commonly involves the terminal ileum and colon, with about 50% of cases showing ileocolic involvement, 30% limited to the small intestine, and 20% confined to the colon. Inflammation is patchy, with skip lesions, healthy areas between inflamed segments, unlike the continuous inflammation and ulcerative colitis.
Okay, let's talk a bit about the morphological features of Crohn's disease.
Crohn's disease shows patchy, transmural inflammation with deep ulcers, cobblestone mucosa, and skip lesions in the gut wall, often in the ileum or colon, visible on endoscopy or imaging. Microscopically, there's granulomatous inflammation with non-caseating granulomas, lymphoid aggregates, and fibrosis, which can lead to strictures or fistulas.
So remind me, when we compare Crohn's with ulcerative colitis, one is continuous, the other isn't, and one has granulomas and the other doesn't. Which is which?
Crohn's disease and ulcerative colitis are compared because both are types of inflammatory bowel disease with overlapping symptoms like diarrhea and abdominal pain, but they differ in their patterns and pathology. Crohn's has patchy, discontinuous inflammation, skip lesions, with non-caseating granulomas, and can affect any part of the gut, while ulcerative colitis shows continuous inflammation limited to the colon and rectum, without granulomas.
How is it that one is not continuous with and the other is continuous without?
Crohn's disease has discontinuous inflammation because it involves patchy immune responses triggered by genetic and environmental factors, like NOD2 mutations or gut bacteria, targeting specific gut segments, leaving healthy areas in between, called skip lesions. Ulcerative colitis has continuous inflammation because the immune attack, possibly driven by different genetic loci and microbial triggers, uniformly affects the colon's mucosal layer, starting from the rectum and spreading proximally, without gaps.
Okay. Interesting. Okay, and let's talk a bit about why is it that we don't have a continuous one with granulomas?
In Crohn's disease, the immune system's attack is patchy.
In general. So we have something called Crohn's, not continuous with granulomas. We have something called ulcerative colitis, continuous without granulomas. Why don't we have one category which is something else, which is continuous with granulomas?
The reason we don't commonly see a category of inflammatory bowel disease that's both continuous and has granulomas is likely due to the distinct immunological and genetic mechanisms driving Crohn's disease and ulcerative colitis, which shape their unique patterns. Crohn's disease involves deep, patchy inflammation with granulomas clusters of immune cells because of specific triggers like NOD2 mutations that cause a scattered, granulomatous response to gut bacteria, while ulcerative colitis involves a continuous, superficial inflammation without granulomas, likely driven by different immune pathways affecting the colon uniformly. A continuous, granulomatous pattern is rare in IBD because the conditions favoring granuloma formation, like in Crohn's, tend to be focal rather than diffuse, and diseases with granulomas, like tuberculosis or sarcoidosis, don't typically mimic the continuous mucosal involvement of ulcerative colitis. Some overlap syndromes or unclassified IBD cases might show mixed features, but a distinct category with both traits isn't well established due to these differing disease processes.
Okay. Let's talk about fat wrapping in Crohn's disease. And cobblestones too, and strictures and skip lesions.
In Crohn's disease, fissuring refers to deep, knife-like ulcers that penetrate the gut wall, contributing to complications like fistulas or abscesses due to transmural inflammation. Let's talk about further upping in Crohn's disease.
Dude, I asked you about cobblestones, strictures, skip lesions, fibrosis, and fat wrapping in Crohn's disease.
All right, let's clarify and cover these features of Crohn's disease simply. Fissuring, not further upping, refers to deep, linear ulcers slicing into the gut wall, often leading...
Wrapping.
All right, I hear you. Let's skip any wrapping up and dive back in.
Dude, wrapping of fat, adipose tissue.
My bad. I think you meant wrapping as in fat wrapping, not wrapping up. In Crohn's disease, fat wrapping, or creeping fat, is when mesenteric adipose tissue grows over the inflamed bowel, especially the ileum, giving it a thickened, encased look, and is thought to contribute to inflammation and stricture formation. Cobblestone mucosa looks like raised, inflamed patches with deep ulcers in between, resembling cobblestones, due to patchy inflammation. Strictures are narrowed sections of the gut from fibrosis, scar tissue buildup, causing blockages and pain, while skip lesions are healthy gut segments between inflamed areas, a hallmark of Crohn's patchy pattern.
Okay. Okay. Let's talk about the microscopic appearances of Crohn's disease. So transmural or full thickness inflammation of the bowel wall. Why would we would see that? Yeah, let's talk about preserved cryptarchic architecture but mucosal ulceration.
In Crohn's disease, transmural inflammation, affecting the full thickness of the bowel wall, occurs because the immune response, driven by genetic factors like NOD2 mutations and microbial triggers, penetrates deeply, causing intense, patchy inflammation from mucosa to serosa. Microscopically, you see preserved crypt architecture in some areas, despite mucosal ulceration, as the crypts, tubular glands in the mucosa, can remain intact or partially recover, while ulcers form above them, alongside non-caseating granulomas, lymphoid aggregates, and fibrosis in deeper layers.
Okay. And what does transmural mean? So what is transmural inflammation in Crohn's disease and what is a fissuring ulcer?
Transmural inflammation in Crohn's disease means the immune-driven inflammation penetrates all layers of the bowel wall, from the mucosa to the serosa, causing deep damage like thickening, fibrosis, or fistulas due to aggressive immune responses often linked to NOD2 mutations. A fissuring ulcer is a deep, linear ulcer that cuts through the mucosa and into deeper layers, like a crack, often leading to complications like abscesses or fistulas because it breaches the gut wall's integrity.
To understand this transmural, tell me the etymology.
The word transmural comes from Latin roots, trans meaning across or through, and murus meaning wall. In Crohn's disease, it describes inflammation that extends through the entire thickness of the bowel wall, from the inner mucosa to the outer serosa, causing deep tissue damage.
And okay, let's talk more about complications. So we might have intra-abdominal abscesses. We might have deep ulcers leading to fistula. We might have a sinus tract. Explain what these things are really, really simply. Yeah.
All right, let's break down these Crohn's disease complications super simply. An intra-abdominal abscess is a pocket of pus inside the belly, caused by deep inflammation or infection from Crohn's damaging the gut wall. Deep ulcers can turn into fistulas, which are abnormal tunnels connecting the gut to another organ or the skin, or sinus tracts, which are blind-ended tunnels that don't fully connect, both forming because the inflammation eats through tissue layers.
Explain this a bit more.
Okay, let's dive deeper into those Crohn's disease complications, keeping it simple and clear. An intra-abdominal abscess is like a balloon of pus that forms in your belly when Crohn's inflammation gets so intense it causes infection, often because bacteria leak through a damaged gut wall, leading to pain, fever, or swelling. Deep ulcers in Crohn's can erode the gut wall completely, creating fistulas. Think of them as unwanted tunnels that connect the intestine to another organ, like the bladder or skin, causing leakage of gut contents, or sinus tracts, which are similar but dead-end tunnels that don't fully connect to another structure, both resulting from the aggressive transmural inflammation eating away at tissues. These happen because Crohn's doesn't just stay on the surface, it digs through all layers of the gut, letting bacteria and inflammation spread, which can mess up nearby organs or tissues.